A macromolecule folds by controlling internal motion. To reach the next threshold of emergence, the focus shifts outward. A cell survives by controlling its boundary. We often treat the cell as a microscopic bag of interacting chemistry, but this heuristic misses the mechanical necessity of its structure. In general emergence mechanics, a cell is a bounded coherence volume. Its survival is the ongoing process of shielding, routing, and regulating spatial gradients. This boundary is never absolute. A completely closed cell dies from isolation. A completely open cell dissolves into its environment. Life exists exclusively in the narrow, highly megulated space between total isolation and structural leakage. This 3D grid visualizes a spatial gradient, a difference in charge, concentration, or pressure across space. Here, the sharp topographical cliff represents an extreme concentration gradient. Unconstrained gradients are powerful enough to drive work, but their uncontrolled collapse produces destructive stress as they attempt to flatten out. At the cellular scale, GEM defines life as bounded gradient maintenance through time. The energetic cost of maintaining this spatial difference is expressed as G-grad, the integral of lambda g times the gradient of c squared, integrated across space. C represents the concentration or charge field. Lambda g is the stiffness required to hold that field, and del c measures the spatial variation, the steepness of the gradient itself. Without a boundary, the cost of maintaining the gradient rises until it is unsustainable. The cliff flattens, concentrations equalize, order washes into the environment. A biological membrane acts as a mathematical constraint. It controls where gradients may form and how they relax, allowing the difference to persist. The membrane's primary systemic function is to lower the energetic cost of maintaining internal thermodynamic difference. When we expand this into a fully enclosed cellular system, the total cost of persistence, G-cell, is defined by 3 specific variables. G-internal is the cost of maintaining order inside the volume. G-boundary is the cost of sustaining the membrane and internal compartments. G-exchange is the energetic cost of controlled traffic across the boundary. The cell remains admissible only while the sum of these costs stays below G-crit, the failure threshold. Equilibrium is the state a dead cell approaches as its gradients flatten and its internal order collapses. Metabolism is the continuous payment required to keep the cell admissible. It is the active process of gradient maintenance, keeping ion balances from collapsing and rebuilding the boundaries that hold them. Death is a sequence of coherence failures. Membranes weaken, gradients flatten, and internal viscosity rises. The cell simply stops paying the cost of remaining itself. Life is chemistry that has become bounded enough to preserve difference through time. A cell is not one uniform space. It is a layered coherent system. Compartments allow the nucleus to protect genetic information, while the mitochondria manage energy conversion, and lysosomes contain destructive chemistry. Each compartment localizes risk and shields a specific gradient, preventing one type of chemical process from destroying another. Many cells maintain voltage across their membranes, a state of coherence tension held across the boundary. Ion pumps spend energy to preserve this difference, while ion channels release it in controlled ways to do work. A nerve impulse utilizes this exact mechanism. It is a traveling change in membrane admissibility, relying on the cell's ability to rapidly release and then restore these gradients. This makes the cell a negotiated city of coherence, where boundaries are used to safely store potential energy for immediate localized use. The cellular GEM triad, depth, stiffness, and viscosity, determines the cell's ability to survive perturbation. Depth provides an energetic reserve, while stiffness ensures the integrity of the cellular boundary. Viscosity dictates the cell's recovery profile, its capacity to return from a disturbance without getting stuck in a deformed state. Too much rigidity causes signaling to suffer. Too little integrity leads to gradient leakage. High internal viscosity slows transport, while a lack of depth makes recovery impossible. Cellular failure is the mathematical inability to maintain this balance under stress. A cell is matter that has drawn a boundary and learned to keep paying for that boundary. This architecture scales to the emergence of mind. It inherits these exact cellular boundaries, membrane gradients, and signaling mechanisms. When we look at a brain, we are looking at a living gradient architecture, built from these cellular networks, bound by the same metabolic costs and recovery profiles. Consider the possibility: is life a special substance, or simply chemistry that has learned how to remain bounded?